So let's start. ELISA, enzyme linked immunosorbent assay, that include two methods, direct ELISA and indirect ELISA. Direct ELISA includes sandwich ELISA and competitive ELISA. Then indirect ELISA. So we may know the direct ELISA, we already learned in the previous class. Direct ELISA is used to detect the antigens, used to detect antigen. Direct ELISA used to detect antigens. Okay, so that indirect ELISA, which means it is used to detect the antibodies. Indirect ELISA used to detect antibodies. Okay, so when we are going to say we have to use it to detect antigens in direct ELISA, so what is required in laboratory? We are going to detect antigen. Their specific antibodies are required. So that we are going to do. So that in that direct ELISA, so first of all, let me introduce the sandwich ELISA. Sandwich ELISA. Okay, so the first we have to go with sandwich ELISA with the diagram. Diagrams will make us to understand everything. Okay, so number one, sandwich ELISA. Okay, so in that we are going to do both positive test and negative test. That we understand much better when we do like that. Okay, so in that the first positive test, let me write it as positive. So we can't say it actually. Before we do the test, how do we come to know the positive and negative? But that for our understanding, easy understanding, we are making heading itself by as positive ELISA. Directly we are putting the heading as positive ELISA. So now let me take the micro titri plate. So will we use the red color here to understand, to see the clear visibility. So micro titri plate. Okay. And next, what we said, in case you are going to detect antigens, isn't it? So that's why what you need to carry here, we need to carry antibodies in the laboratory. We need to carry antibodies. I'm going to draw the antibodies which are attached to the well. Antibodies attached to the well. Okay. So what we do then? So first the sample number one, that we need to write the notes also along with that. I am writing notes that first plate. For that, what we'll write? Antibodies of laboratory. That for our better understanding, I am writing antibodies of laboratory which can bind with specific antigens, which can bind with specific antigen are attached to the microtitry plate. Okay, antibodies of laboratory which can bind with the specific antigen are attached to the microtitry plate. Okay, next the second box. What we do? We are going to add that patient sample. Number two, patient sample is added. Shall I change color? What will be better for us? Patient sample is added. Okay, so patient sample is added. So when we add the patient sample, if antigen present, in the patient sample, if antigen present in the patient sample, if we have to say, because we can't see, if antigen present in the patient sample, they bind with, they bind with antibody. They bind with antibody. Okay. If present, they bind with the antibody, which I am showing here. They present and they are binding with the antibody. So they are present and they are binding with antibody. So Antigen I am showing here. Antigen is there and they are binding with the antibody. Okay, that is the second one. And in the third, what we are doing, another antibody is added to the microtitry plate, another antibody. Okay, so here to make a differentiation, primary antibody I am using here, a word I am using, primary antibody, another primary antibody added to the microtitry plate. That means whatever we added earlier also, that is going to be that primary antibody. So what is primary antibody? Let us know the secondary antibody. You'll find what is primary antibody. So another primary antibody is added to the microtitry plate, and it will go and bind with the antigen. If it is present, it will go and bind with the antigen. So I am writing antigen, I am showing antigen first of all. So making antigen here. Next, we need to add that primary antibody, another primary antibody, which is binding with the antigen on other side. Another primary antibody is added, and that is binding with the antigen on other side. Okay, this is not even enzyme linked. Okay, so next. Then what we have to write? If antigen present, this antibody, this primary antibody attached to the antigen on other side, attached to the antigen on other side. Okay, if antigen present, this primary antibody attached to the antigen on other side. Okay, next, what is the fourth one? That what we have to do then. But let me show you that all the things which is happened till the time. We have an antibody which attached to the well, and that we have antigens attached to the antibody if antigens are present. And then next we are taken another primary antibody which is going to bind with the antigen on other side. Now what we have to do? Enzyme linked secondary antibody is attached. Enzyme linked secondary antibody. Enzyme linked secondary antibody is attached. Added. First of all, we have to add. Then after, if it is, if they found primary antibody, they go and bind with them. Enzyme linked secondary antibody is added, and it binds with, primary antibody. What is secondary antibody? Secondary antibody means the antibody which can attach with the antibody. The antibodies who are produced against another antibody, we can say secondary antibodies. Okay. So these are also available in the laboratory. That now they are going to be enzyme linked. Secondary antibody is going to be enzyme linked. Okay. So secondary antibody. Let me say you that secondary antibody, how we prepare. That if you want an antibody against the antibody, that seems to be the word seems to be a little bit confusing. That if you want an antibody against the antibody, let's take a mouse or mice here. So if I introduce an antibody into the mouse, what happens? Mouse feel it as antigen, isn't it? So then it produces against antibodies against an antibody. It is antibody for you, but not for mouse. Did you get my point? It is an antibody for you, but not for mouse. So that's why if I introduce any antibody to the mouse, so what it will do? It will produce a new antibody that we call secondary antibody. That we say secondary antibody. So just to be remembered. So finally the four we got, and the fifth one I am showing bigger because that is the positive one. The fifth one I am showing bigger. That let me show you the full form of this. the microtitary plate. Now the condition is going to be that an antibody, primary antibody who attach to the well, and next that antigen if present, antigen if present, that attaches to the another that to the antibody. Next, another primary antibody is added that goes and attaches to the antigen on other side. Okay. Next, enzyme linked secondary antibody is added. Enzyme linked secondary antibody is added, and that go the that antibodies who can bind with an antibody, the antibodies which are produced against an antibody, we call it as secondary antibody. So how do they prepared over? I said it now. Then let us label all. Let us label all. That means if we label with this single diagram, we can understand anything. Number 1, number 2, 3, and then 4. Okay. So what is the shown one? That is secondary antibody, which that is enzyme linked. We are linked with enzyme, that secondary antibody. Secondary antibody is enzyme linked. Okay. Next, this is primary antibody who binds with an antigen on other side, and this is the antigen. This is the antigen. This is the key one. So that if you understand there is the patient sample, if there is an antigen, then only all this binding is possible. Okay, so that is an antigen. Next, the one more primary antibody. One more primary antibody is there which attached to the well. Okay, so and the between every sample we are going to add that which is the free fluid absorption is very common. Okay, so then now add substrate. So add substrate, I am writing here, add substrate. So then when we add substrate, the substrate which can bind with, the substrate which can bind with this enzyme. Okay, so when we add substrate which can bind with that enzyme which is coated on the secondary antibody, that makes a change in the color. That makes a change in the color. The reaction in between them can change the color of the microtitary plate. If the change in the color, if you see what you can understand, that antigen is present in the sample. Antigen in the sample. So change in color. So declaring here, change in color. What you can understand if the change in the color? Substrate reacted with, substrate reacted with, the substrate is reacted with enzyme, that you can understand. So that means when substrate is reacted with enzyme, what we can understand? Enzyme linked secondary antibody present. Enzyme linked secondary antibody present. That means enzyme linked secondary antibody is present means it attached to the primary antibody. That means it attached to the antigen. That means we identified the presence of an antigen. We identified the presence of an antigen. Okay. Then what we do here, that let us do the negative test. Then we can understand much better. Negative test. So the incompleteness will be completed over when we do the negative test. Okay, the second one, the negative test. We should not announce actually before itself, this is positive test, this is negative test. How can we say that when you go to the laboratory, what he do the test positive or negative, whatever. But here we are learning, that's why I'm showing this kind of variations. So let me draw the similar kind of microtitary plates as usual, like this. I drawn 4 earlier, and the same I'm doing here. As usual, that we have to say there is an primary antibody attached to the well, that microtitary plate, the well of microtitary plate, the primary antibodies are attached. Then next, what we have to do, the patient sample is added. If antigen present, they bind with the patient sample, but there is no antigens maybe. So that's why the no nothing is actually not at all. Everything is anything is not visible here. Just for our understanding, we are saying. If patient sample contain antigen, they go and bind with the antibody. Okay. Next, what we have to do the next that what we have to add, the another primary antibody is added here. Another primary antibody is added. Next, the fourth sample, what we have to add, enzyme linked primary antibody is added. Enzyme linked, the fourth sample, what is added, enzyme linked primary antibodies are added. So shall I change color for that? Enzyme linked primary antibodies are added. Okay, so for this, I have to write the notes. Then after we'll go with number one. What we said about the number one, that antibodies of laboratory attached to the microtitry plate. Antibodies of laboratory. Antibodies of laboratory. Which antibodies? They are primary antibodies. Antibodies of laboratory attached to the. It is right. Primary antibodies of laboratory attached to the microtitry plate. Okay, these are of laboratory. Next, the second one. Patient sample is added. Patient sample is added. Okay, if antigen there in the patient sample, if antigen in the patient sample is present in the patient sample, same we have to write. We don't know actual present or not. If antigen present in the patient sample, they bind with antibody. They bind with antibody. If antigen present in the patient sample, they bind with antibody. Okay. Next, the third one. Another primary antibody is added. Another primary antibody is added. Okay, so another primary antibody is added. If antigen present, they bind with antigen on other side. If antigen present, they bind with antigen on other side. Okay. Next, the fourth one. The fourth one. So why the in the third diagram to the fourth diagram, why you can't see these antibodies? Why we can't see these antibodies in third to the fourth? That's what I said over. In every level to the next level, we are going to absorb the free fluid. When you absorb the free fluid, that will go. So next, the fourth, the enzyme linked secondary antibodies are added. Enzyme linked secondary antibodies are added. Okay. When we add enzyme linked secondary antibodies, what we have to write? If if primary antibody present, they bind with them. Should write. If primary antibody present, they bind with. Okay, not for this antibody. That the another primary antibody we are talking about. If primary antibody is present, who is they bind with them. We should write that. They bind with them. If primary antibody is present, they bind with them. So this is the story. Final negative story with the bigger diagram. So negative test. What you'll find? You will find only what we added in the beginning. Means if there is no antigen in the patient sample, no primary antibody attaches to that antigen. And no primary antibody present means no enzyme linked antibody attached to that particular antibody. So finally, after all watching the free fluid, you will find like this. Then add substrate. Add the substrate. What happens if you add substrate? Add substrate. which can bind with the enzyme. But there is no enzyme actually. So how we come to know there is no enzyme? No change in the color. No change in the color. Okay, if there is no change in the color, that we can understand. If there is no change in the color, then we can understand the test is negative. Okay, so finally, that positive case, the test that scene will be like this. That you have to remember. Seems to be so many diagrams we run, but only one diagram positive, one diagram negative. Then when the color is changed, we can identify the presence of this secondary antibody. We can identify the presence of secondary antibody. Secondary antibody present means that attaches to the primary antibody. That is present means antigen is present. Antigen present means that disease is detected. In that way, you have to understand. If there is no change in the color, if there is no change in the color, okay, no change in the color means no enzyme linked secondary antibody. That means no another primary antibody. That means no antigen. No antigen means the test is negative. That antigen is not detected in the patient. So this is Sandwich Elisa. This is Sandwich Elisa. About the direct Elisa, that in the direct Elisa, we are learned the Sandwich Elisa positive test, Sandwich Elisa negative test. We learned. So then after we have to go for competitive Elisa. We have completed this Sandwich Elisa. We have completed. So then after we have to go for that competitive Elisa. Competitive Elisa. Sandwich Elisa of direct Elisa we are completed. Okay.